Hey dear learners, I again welcome back to the second part of the previous video. Okay, because the previous video was too long, so I decided that in order to make you feel a bit easy, okay, to understand the topics in more depth, so I decided to continue the second part of the previous video. Okay, now this part will be regarding the outbreeding devices, pollen crystal interaction, and the artificial hybridization concept. Is that clear? Without any delay, let's get started. Okay. Look, what is outbreeding devices? Okay. Look, first of all, outbreeding devices is generally like thing, a process to promote cross pollination instead of self pollination. Because why they do so? Because continued self pollination result in inbreeding repression. Okay. Done. Okay. Now look what is given from very first. Majority of the flowering plant produce hermaphrodite flowers and pollen grains likely to come in contact with the stigma of the same flower. Now what is being told? Hermaphrodite. Now what is hermaphrodite mean? Hermaphrodite means bisexual flower. Okay. Many of the flowering plants are bisexual only. Okay. So hence a lot of chances are possible that they do self-pollination only. Okay. So in order to prevent them, you are creating certain outbreeding devices. Okay. And I have already told that what is the drawback of outbreeding devices because self pollination will lead to the inbreeding separation. Okay, means I think you have already in practical in human case you know already that marriage should not be done within the same family. Okay, it should be done in families that are not very close, very distant family. Okay, the marriages should be done in case of humans so that any kind of genetic deformation not occur to the child. Okay, that is the main reason. Similarly, in flower also, self-pollination will lead to the inbreeding self-depression. So that is the main reason why the outbreeding devices are there in order to promote cross-pollination. Where where the self-pollination is the maximum chances of self-pollination is maximum. Okay, so flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. Now, in the look, the yellow points that I have highlighted, they are nothing but what they are the outbreeding devices types. Okay, number one will be like pollen release and stigma receptivity are not synchronized. Okay, means the time when the pollen grains and the stigma released. Okay, time period will be very different. It is not homogamy, it is heterogamy. Okay, that is the main thing. If heterogamous happen, then definitely chances of self pollination is very negligible because the pollen grain released will not be able to meet what the stigma of the female part. So fertilization cannot occur. That is the main thing they are telling here. Okay, either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the release of pollen. That is the main thing they are explaining. Okay. In some other species, the anther and stigma are placed at different positions. Yeah, definitely. Um, I think I'll be teaching you in artificial hybridization the concept of emasculation, bagging, etc. But that is completely different topic. I also know. But look that anther and stigma please separated from each other. Okay, so that pollen cannot come in contact with the stigma of the same flower. Clear? Okay. Both these devices prevent autogamy. Now, autogamy in the previous video, if you have not watched, do watch it. Yeah, do watch it because if you don't watch the previous video, okay, you cannot understand the concept of autogamy in detail, where I have taught you genetic and physiological happenings that happen. Okay, so two devices we have done where heterogamy or the release of pollen and stigma are not synchronized. Done. The second we discussed was that the anther and stigma at different positions. Two devices done. Third device was self incompatibility. It is mainly related to this, not this, but a genetic mechanism. What is self incompatibility? It is a genetic mechanism that prevents cell pollen from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. But I think my English, okay, from my English, what I've read it just now, I think you guys all made out the meaning. Okay, there is a concept of self incompatibility. Okay, what is the fourth device? Production of unisexual flowers. Now we have already discussed that in majority of the flowering plants, they are hermaphrodite. Now this time what will happen? They will be producing unisexual flowers. Okay. So if both, this is only given. Okay. Now look, you will get two concepts. First of all, first concept will be monoecious, and the second concept will be dioecious. Look, I'll be telling you it very detailed, minute detail. What is monoecious? Monoecious, it is a bisexual plant, but it can possess unisexual as well as bisexual flowers. Each example will be each example will be castor maize. Okay. So what happens in monoecious? Monoecious prevent autogamy but not jatonogamy and xenogamy. Okay. It prevents autogamy because the pollination cannot happen within the same flower but happens in the same plant. Okay. That is the main thing. Because unisexual flowers. Yes. For autogamous to happen, it should be bisexual plant with bisexual flower. Okay. Yeah, bisexual flower that too close, clistogamous flower. But what is happening? Bisexual plant, unisexual flower. Okay. So hence preventing autogamy. Clear? Now, what are the examples? I have told you about autogamy, castor and maize. Now moving ahead with dioecious flower. What happens in dioecious? Plant is unisexual, flower is also unisexual. Example, paya. So as a result, it is preventing both autogamy and jatonogamy. Unisexual flower is preventing autogamy, and jatonogamy is being prevented by what? Unisexual plant. Simple. Is the concept clear? Listen to me, whatever I have taught. Okay, just spoken just now. Okay, just go back, listen it one or more, two more times to make it concept very clear. Okay, I am reading it out here. What is given? It prevents autogamy. Okay, female flowers are present on the same plant. If both male and female flowers are present on the same plant, such as castor and maize, it becomes monoecious. It prevents autogamy but not jatonogamy. In several species papaya, male and female flowers are present on different plants. That is, each plant is either male or female dioecious. This condition prevents both autogamy and jatonogamy. Is the concept clear? In monoecious, bisexual plant with unisexual flower. Clear? Example, castor maize. What happens in dioecious? Bisexual not. Bisexual, no bisexual term in case of dioecious. Okay. Unisexual flower and unisexual plant. Example, papaya. Concept clear? Done. Now look, in the pollen pistil interaction. Okay, I have not highlighted. I will make the concepts to you very clear. Okay, first of all, look. Pollination. Okay, what is given? Pollination does not guarantee the transfer of right type of pollen. Means, what is right type of pollen? Means, whenever, because it is a chance factor, okay. Suppose the pollen grain released from the male is of hibiscus, okay. But the released pollen grain, okay, is being carried by wind, water, or insect to another plant like rose. So is the pollen of right type? Hibiscus and rose? Definitely not. They are different species. Yes, different flowers, different functions. They will not definitely match each other. Okay. So pollination by means does not guarantee the transfer of right type of pollen. Is that clear? But however, okay, you listen to me now very carefully. If the pollen grain released, okay, of compatible type. What does compatible mean? Means same type, like hibiscus male, pollen grain landing on the hibiscus stigma. Okay, so it becomes what? Compatible type. So if pollen grain is of compatible type, pollen grain will germinate and give rise to pollen tube through one of the germ pores. Clear? Pollen grain will germinate and give rise to pollen tube through one of the germ pores. What will happen next? The content of the pollen grain. Okay, means what? What is the content in the pollen grain? Pollen grain, if it is set at the two cell stage, it will contain a generative cell and a vegetative cell that I taught you in the male part. Yes, remember pollen grain structure that I was discussing male gametophyte part. Yes, so content the pollen grain. If two cell, then two cell only, two cell stage, generative and vegetative. If it is a three cell stage, means generative, vegetative, and the generative cell being divided two part into two male gametes. Okay, the content of the pollen grain will definitely now move to the pollen tube that germinated. Okay. if the pollen grain landed on the stigma of the compatible type. Clear? The two steps clear. Pollen grain, if it is of right type, compatible type, it will germinate. Pollen grain will germinate and give rise to the pollen tube through the jump pore where exine was absent. Now, the content of the pollen grain will move into the pollen tube. Clear? Done. What will be the third one? Third step will be pollen tube continue growing through stigma and style until it reaches the ovary. Clear? Pollen tube now will elongate until it reach the ovary. And from which end it will enter? It will enter from the salazar end, I think. Okay, where the synergies were mainly present. Okay, so synergid part where it is present, from there only it will enter. Okay, it will not enter. It will not enter from the antipodal side, but it will enter from the salazar, not microbial end. I think yes, microbial end. Okay, let me look back because I have taught you that MS is AC. M means what? Microbial synergies. So microbial energy. Yes. Just remember the trick. Microbial MS is AC. Mahendra Singh Dhoni is cool. What is cool? Cool other name is AC. So for this like this short tips, okay, in your NCERT completely biology, complete NCERT biology, do subscribe to our channel for further information, for further tips, tricks, etc. Whatever you say. Okay. Now look, pollen tube will continue growing. Okay, and it will reach the ovary. Is that clear? Done. Okay. Now what happens? Pollen tube reaches the microbial. Why it reaches the microbial? Why did not enter through the antibodies or salazar? Because the microbial end consists of what? Filiform apparatus, which are nothing but what? Cellular thickenings, which is the main region of guiding the pollen tube through them. That is the main region. Okay. Now after it reaches the microbial end, okay, pollen tube absorb water from the synergids, and as a result, pollen tube swells. As it swells, it bursts out, and the two male gametes are released. Is that clear? Now, after what happens, we'll gonna discuss in double fertilization part. So, all the events clear? Clear? Pollen tube, pollen grain, if right type, germinate, pollen tube grow. Now, pollen tube, all the contents of the pollen grain will be given to the pollen tube. The pollen tube will go down till it reaches the microbial end. Why it enters through the microbial end? Because of the presence of the filiform apparatus, okay, which ultimately guide them through the microbial end. Now, after it receives the microbial end, it absorbs the water from the what synergids, burst out, and two male gametes are released in the ovary. Is the concept very clear? Yes, I feel it is clear. Now you can read all this topic, whatever I taught you. Okay, these lines, whatever is given, very simple. Whatever I taught you, whatever I told you, everything like that. Concepts clear, everything is done, everything is sorted. Okay, let's go back. Okay. The ability of pistil to recognize the pollen followed by its acceptance or rejection is a result of continuous dialogue between pollen grain and pistil. So now how they judge there is of right type or wrong type, it is mainly given. Okay, it is mediated how chemical components of the pollen interacting with those of the pistil. There is the main thing they are telling, highlighting. Okay, you can also highlight in the NCERT. Okay, I'll move ahead. It is only in recent years that botanists, okay, nothing else. As mentioned earlier, following compatible pollination, the pollen grain germinates to produce a pollen tube. Yeah, I have already told these things. Two cell stage pollen tube after reaching the ovary. Yeah, I have already mentioned earlier. Okay, you just listen to me very carefully, make notes of the point. Everything is done from this topic. All these events from the pollen deposition on the stigma, man, okay, is referred to as pollen. So, whatever I taught you, whatever I spoke to you just now, all the events, okay, please make a note of them. Write in your board exams wherever you want. You will get full out of 4 marks in pollen crystal interaction. No need to mug up these concepts. Okay, not concepts, no need to mug up these lines. Okay, because very difficult lines they give NCRT. It is very difficult to understand. Okay, so whatever I told you, if you made your concepts clear from my uh, speaking part, okay, then definitely is gonna create a great difference between you and your classmates. And if anything you have doubt, regarding pollen pistil part, okay, please mention in the comment section. I am always there to help you out. Okay. Pollen deposition the stigma until okay. Now, what is pollen pistil interaction? It is a dynamic process. Okay. Involving pollen recognition followed by okay, everything I have done. You can easily study pollen germination by dusting. Okay, you do in the practical class 12 practicals. Okay. Now coming to the concept of important concept called artificial hybridization. This is a very important concept. Okay, in today's era, because either a question will come from outbreeding devices or a question will come from artificial hybridization from today's video. Okay, the question obviously can come from the pollen bristle interaction. Only you need to write the concepts. No need to mug up like anything. Okay, you write the concepts whatever I spoke to you. Okay, you write them point by point. Everything is done from pollen bristle. Okay. Now moving ahead with artificial part. Okay, artificial hybridization. Okay, what is artificial hybridization? Artificial hybridization is nothing but it is a crop improvement program. What is it? It is a crop improvement program where desired pollen grains are used for pollination. Okay. Now certain steps of artificial hybridization. Step number one will be selection of desired parents. Okay, means it is a case like hybridization. Okay, for example, you want to produce milk. Okay, good quality milk with disease resistant behavior. So you are gonna mix two varieties that are very superior in them. Yes. Similarly, it works in artificial hybridization like that. Okay. What is the first step? Selection of desired parents. Second step, emasculation. What is emasculation? Emasculation is removal of anther from floral bud to prevent self pollination. Is that clear? It is the process of removal of floral bud to prevent self pollination, and it occurs. See in a pink highlighted part. If the female parent bears bisexual flower, we only need to do emasculation. If the female parent produces unisexual flowers, there is no need of emasculation. Is the concept clear? Then we'll move on to the third step called as bagging. What is bagging? To prevent unwanted pollination, pistil is covered with butter paper. Okay. To prevent unwanted pollination, pistil covered with butter paper. This is the bagging part. Then what? Collection of pollen from male parent. Okay, fourth step. Fifth step, dusting pollen grain on stigma. And last step will be rebagging. So these are certain steps. These are the 6 steps that defines artificial hybridization. Steps involving artificial hybridization. Step number one, selection of parents with desired pollen grain. Not desired pollen grain, desired characteristics. Okay, what is step number one? Selection of parent with desired characteristic. Number second, emasculation. Emasculation, only case of bisexual flowers, female, obviously. Okay. If it is a unisexual female flower, no need of emasculation. Then third will be bagging. Fourth will be collection of pollen from male parent. Number fifth will be dusting of pollen on stigma. Number sixth will be rebagging. Okay. So this is all about artificial hybridization. The points I have highlighted given, and if you want, okay, you can read them in detail. But you will not get anything new. Okay, I can assure you that whatever I spoke to you, that is the only thing given out here. Okay, so I think that ends the part 2 for the previous video, because it was previous video will be, if I have included these points in the previous video, it will be very hectic for you to follow that. Okay, so it will be a very huge cost duration. So I don't want to create burden on you all guys. Okay, so again, you guys made it to the end, and I feel you will make the series very successful. Till then, keep practicing, stay curious. Thanks for watching. If you have any question, leave them in the comment section below, and do hit the subscribe and the like button too. Thank you all guys.